Hello, everyone, and welcome to today's live broadcast, Harnessing the Power of Healthcare Data with Causal Machine Learning to Drive Personalized and Effective Treatment Interventions, presented by Dr. Aya Khalil. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's presentation. Today's educational web seminar is presented by LabRoots, the leading scientific social networking site and provider of virtual events and webinars, advancing scientific collaboration and learning. We'd also like to acknowledge the C&EN Media Group for help with programming and promotion of this event. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Please also notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble hearing or seeing the presentation properly, please click on the support button at the top right of the presentation window or use the Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process for obtaining your credits. I'd now like to introduce today's presenter, Dr. Aya Khalil. Aya Khalil, the co-founder and executive vice president of GNS, has promoted the use of the company's technology in the biopharma industry. One of the inventors of the company's computational platform, her work has helped major players such as Biogen, IDEC, and Johnson & Johnson to make computer-driven discoveries for the respective R&D programs. If you would like more information about our speaker, please click on their photo image in the top right. Please join me in welcoming Dr. Aya Khalil. I'll now turn it over to her for her presentation.
Hello, and thank you for the invitation to present today. It is my pleasure, so I'd like to thank the organizers for this opportunity. And as Brenda had said, my talk today focuses on the use and how we leverage machine learning, causal machine learning, to drive personalized medicine. So big data has become now a big buzzword in our industry, especially for those of us who've been in this for a while. I think there was a time when we used to call big data by another name. It was called systems biology. And what it was really about was enabling biologists, folks in the medical field, healthcare field, to go beyond just being able to observe one factor, but looking at many, many different variables and factors. So being able to sequence whole genomes, observe transcriptional levels at scale, at the genomic, microRNA, proteomic, metabolomic, and link this all the way to the phenomic, our ability to observe how the granular genetics, microRNA can actually impact things on the phenotypic and physiological level. And 15 years ago, these technologies were just starting to emerge. Now they're becoming more ubiquitous and are shedding light in systems, whether it's biological, medical, or healthcare, in ways that we couldn't even imagine. So, but with all this data comes a challenge. And the challenge is, how do we actually make sense of this? So we can go into the random news at any given day and learn about how, our propensity for drinking coffee can then potentially lead to a myriad of disorders, which may or may not cause various effects. So we're faced with all of this data. This data can be seen as a set of patterns or correlations, but really our goal is to go beyond these patterns or correlations and learn what causes what in the system. And by getting at underlying causality, hopefully we can get to insights that we couldn't get to before with the data that we have access to today. So we're faced with this perfect opportunity of an exponential rise in data and really our ability to collect it at scale at costs now that aren't prohibitive, massive number of increased and interventions, and breakthroughs in machine learning and cloud computing. And our goal, or my goal, has been really developing algorithms that can go beyond what we already know, what's already established in the literature, and algorithms that answer what I like to call the what if. If we can get at causal mechanisms, then we can start to ask and not only predict outcomes for patients, but what is the right intervention. And for those of you who are on the line that would like to get some of these slides and materials, please feel free to email me at the end of this presentation, and I would be happy to share this with you. So one way to look at machine learning and predictive modeling, this is a pyramid that was reported by Davenport in Tom Davenport in one of his publications and books. You know, we can think of taking data in and doing just standard reports, statistical analyses that measure or try to predict, okay, what's up, what's coming, what's down, forecasting, and even predictive modeling that looks at what will happen next. Our goal is to go beyond predictive modeling and to learn from the data and optimize around the data, learn directly from the data, and infer new mechanisms, new effects beyond what's directly in the data itself, answering the question of what will happen if I do this. So the underlying mathematics that we chose to explore and leverage is what we call the mathematics of causality. So we can look at a data set and start to look at what are the patterns of correlations. And we can ask, does A correlate with B or B correlate with A? And we can even try to attach arrows to those correlations and ask, is there a direction to it, a causal association? And it turns out that mathematically, with just two variables, A can correlate with B, and you can even ask, does it cause it? And you can reverse that association, but you can't distinguish between these two graphical associations, these two graphical models. When we can introduce an exogenous variable, a third variable, and in biology and medicine, that variable can be a genetic factor. It could also be a drug or an environmental factor. That's where mathematically we can probabilistically start to discern, can A cause B or B cause A? And C in this case can represent in your system, in your data, any intervention that's driving variation in your data, in your outputs, be it on a molecular level or phenotypic or physiological level. And mathematically, these two probability of these models given now any data set that you might be comparing it to are different and will score differently given this exogenous variable. And so we can start to sort out, is A what's driving B in my system and in my data that I aim to study, or is it B driving A? There are also ways to discern and try to come up with patterns of probabilistic causality when you don't know what the exogenous variables are, especially in the context of observational data. And one can then start to model and look at graphical structures that would test, does A drive C or B drive C? And if A and B are weakly associated with each other, the more and more that we start to learn and pick up these kinds of graphs, we can start to sort out, even within the context of observational variable data, from patterns of correlation, causal associations. So that's the underlying mathematics. And our goal was to take that math now and leverage it at scale. So we want to be able to do it for any kind of variable, any kind of data set. Because when we talk about predicting outcomes, it's going to be a combination of measures and variables that allow you to model what's happening in the system, learn directly from data, the relationships between these variables, and we want to do it at scale. And so what we set out to do was really take that mathematics, that underlying mathematics that I had just described, that has been recognized via Turing Awards and developed by folks like Judea Pearl, which have established the mathematics of causality. We've encoded that now into sets of algorithms that can take in raw data and unlimited kinds of data so that you can learn the interactions directly from your data sets, the data that you're feeding into the algorithm. You want to be able to do it quickly. And what we end up learning here is not just a single model, probabilistic directed graphical model underlying the data, but we learn ensembles of models, because there could be more than one model that could actually represent your data. And in learning these ensembles, that's one way to guard against overfitting, as well as capturing the uncertainty underlying your data. And then once we have these ensembles, we want to run simulations and make very specific predictions down to the individual, person by person, member by member. So in a little bit more depth, So what I described is the process now of taking in data that might be diverse, it might be massive, multimodal, of different kinds, of different types. We're now going to run this through an engine. The first phase of that engine is what we call the reverse engineering, the learning of the probabilistic causal associations underlying your data set. And the first thing you need to do is be able to score these models. We're going to use a probabilistic Bayesian framework for scoring these models. The motivation behind that is to be able to sort out these directed probabilistic acyclic causal graphs, but also the Bayesian framework is one way to guard against overfitting, because you're always fitting to the least, most complicated model that is most likely to represent your data. And then we go through a process of optimization. We're going to now parameterize and seed the model with different kinds of networks, and we're going to evolve them as a function of temperature. And we're going to keep evolving them until we end up with the optimal set of models, ensemble of models, which represents a sampling of the possible models that could fit your data. And in that sampling, once we have that, now we can run simulations and ask very specific and targeted questions. What would happen if I were to condition this model on a genotype that matches a specific patient, and I were to modulate a therapeutic target or pathway within that patient, downregulate, how would that impact the outcome? And the answers will come out as distributions. Those distributions reflect uncertainty in the models that are generated, and that's okay. We want to capture that uncertainty, and we want to use that uncertainty to say, this distribution, when I upregulate that target, that drug, that pathway, is very different than that one, and I can say with some confidence how likely that is to impact an outcome measure in a specific case. So that's the process end to end. And what I want to do next is really go through now where we are applying this, how we're applying this, and what this platform around reverse engineering probabilistic networks and probabilistic causal mechanisms underlying your data, and then running simulations to ask what-if questions. Where have they gotten traction, and how we've been able to use them to hopefully advance our understanding of mechanisms driving disease, as well as our ability to predict subpopulations and biomarkers of response, and maybe eventually apply this in a point-of-care setting. And so one of the things that has emerged, as I described earlier, with our ability to measure more and more, is now we can actually run large-scale studies that combine the measurement of genetic, molecular levels, different flavors of molecular variables with clinical phenotypes, producing large sets of codes over hundreds and possibly thousands of cases. And we're working with and collaborating with various entities, from foundations like the Cure Huntington's Disease Initiative Foundation, the Multiple Myeloma Research Foundation, the Inova Hospital System, and Global Genomics, to really leverage these data sets and identify what we can learn from them. And more and more increasingly, with the advent of electronic medical records, we can also apply these algorithms to EMR data sets. So this is an example of work that we conducted leveraging the REFS platform, the reverse engineering and forward simulation, to learn models that predict the transition from normal diabetes to prediabetes to full diabetes. For those who are interested in delving into this study more deeply, this work has been published at the Journal of Diabetes Science and Technology, and you can access it there. But I won't spend too much time on it, so I'd like to spend a bit more time on the data sets and studies that leverage the deep phenomic and genomic studies. But the point being that now, sort of with the ability to collect different kinds of data that are matched to different questions and problems, from real-world data sets to omic, to combinations thereof, and you can combine that now with this capability to reverse engineer these models directly from data and then run your own simulations, it's really fueling different applications and solutions that span the discovery of new biology, biomarker discovery, and even applications in the real world around comparative effectiveness and population health. So now I will go into a bit more depth around a couple of these case studies in the time that we have. So I'll start with the work with global genomics. This is a recent study. We aim to, at some point in the coming months, publish these results, but I thought it's useful to see how we thought about really leveraging this large-scale data set to get at causal mechanisms underlying cardiometabolic disease and cardiovascular disease. And so it always does start with the data. And in this study, this was designed to generate large data sets that we aim to mine and generate comprehensive disease models to come up with potential new targets, biomarkers, as well as apply in a patient diagnostic and treatment setting. And so that's the motivation. And so to do that, the study focuses on 7,500 patients, and different trials are being run within that 7,500, starting with a pilot study where the data set is divided into a discovery and validation cohort, and then the cohort is expanded. And again, we set this up as a discovery and validation cohort, and then the cohort is expanded again to do additional discovery, and then final validation. And the key here is that in this design, we're cognizant of the need to be able to have a phase of training these models and validating them, expanding the cohort and adding in more data on more patients and validating those, and then finally having our final discovery set and validation. And that's built into the design of this study from the get-go. And then on each of these patients, we are measuring cardiac CT and imaging analysis. A lot of this work started with methods around the phenotyping and getting quantitative phenotypic measurements. And then whole genome DNA sequences are measured, along with RNA transcriptomic and sequence data, as well as proteomic and metabolomic. So it's a multi-omic study where DNA variation and sequencing is sort of seen as the anchor by which we're going to start to tease out these causal mechanisms and connect them to the endpoint, to the phenotype that was measured in the study. So that led to, and we're now in the pilot phase of learning and validation, 8 data modalities measured in each patient. We took those 8 data modalities and ran through an exercise of normalizing the data, checking for quality control, and filtering for the variables that actually show signal in the data. So that led to us now focusing our modeling effort and the data frame that we entered into the reverse engineering form of simulation on 103,565 variables. We ran those variables across then those hundreds of patients and reverse engineered models. The endpoint of that reverse engineering exercise was a model where we had to explore over 10 to the 18th network fragments. That exploration was enabled by running the algorithms on the cloud and parallelizing them. And we ended up sampling 6 times 10 to the 11th networks to come up with our final ensemble. And then once we had the final ensemble, we ran pairwise calculations to identify what could be driving what in the system. So we're looking for variations and relationships between phenotype to a molecular measure. That molecular measure can be a blood-based biomarker, a protein marker, RNA, microRNA, etc. To learn the models, because this is a large-scale data set, many variables, it was 6 days end-to-end runtime on a compute cluster. So in running these models, we applied certain constraints. So even though we're looking for and trying to learn the causal probabilistic relationship directly from the data set, we do apply some biological constraints. So DNA variants can drive variation in any one of these molecular outputs, as well as clinical parameters. This could be something like age or gender. And then within the molecular variables, they can drive each other. And then ultimately, they drive an endpoint around cardiac plaque. We also had other endpoints represented in this model. Then once we learned the models, we're now going to probe it and ask, what could be driving what? And really what we're going to do is run a simulation, a what-if query that conditions these models on a specific patient background. and goes through and upregulates and downregulates every node of the model and asks, how does that lead to variation in the endpoint, and do I see a strong change in the endpoint when I modulate any one of these data points here, any one of these genes or nodes. So as I had mentioned, we've just completed these results. We do plan to publish on these, but so far we're really finding a lot of interesting biology in these models, a whole set of novel targets associated with coronary artery disease have emerged, microRNA targets, protein targets, and we've also found and discovered within that the subnetworks that already underlie established biology, the submolecular and genomic networks around LDL biology. So to date, with this approach of really thinking upfront about how we want to design our study, be it an observational study or a clinical study, where we want to go through an exercise of measuring all of what we think are the relevant components to be able to recapitulate and learn these disease models, as well as setting up an experimental design that supports training and validation. We've built to date what is one of the largest molecular and genomic models underlying cardiac artery disease. We've identified known mechanisms within this model, as well as novel mechanisms, and even mechanisms around known targets, but where we want to be able to learn and establish more of the underlying biology. And now we aim to apply this framework and these platforms to generate other predictions around specific patient predictions, guided treatment pathway, further biological discovery. I will continue. I'm going to go through another case study in the time that we have, and this is work that has been published as a poster presentation at the ASH meeting in December, and it is in collaboration with the Multiple Myeloma Research Foundation. And our goal here was to build causal disease models underlying multiple myeloma, and then to make those models accessible to a group of researchers that are associated with multiple myeloma and the COMPASS study. And so the idea here is we're going to create and design a registry that allows us to learn from these data sets, causal networks underlying multiple myeloma. Our focus here was on newly diagnosed patients. So everybody in the study is a newly diagnosed patient, and then they're being followed longitudinally. Their molecular levels are being measured along with SNPs and a whole host of clinical data against the tumors that were collected. And the patients are in different treatments around IMiDs, Revlimid, et cetera. Okay. So we took in that data and built models underlying the system and then interrogated them. And what we've set up here is a process of learning and confirmation. So every 6 months or so, the registry data is enriched with more patients. We take in that additional data and upload it into the BRASS platform to update the model. And we end up learning new links and associations because we have more data and more insights. And we do make these models available to members of the consortia so that clinicians can go in and access it and ask their own what-if questions, come up with their own queries, and generate their own hypotheses. And I'll show what that looks like. So in this case, to reconstruct these Bayesian networks, these are the results from the first data refresh. We have continued the study, and we're working on other data refreshes. So we have about 800 patients. Again, starting from that raw data, we ran QC measures along with normalization, et cetera, to come up with about 28,200 variables that showed variation in that study, and that's what the modeling is going to focus on. And those variables consisted of RNA-seq, SNPs, structural variants, copy number variation, as well as clinical data outputs, things like a first response variable, progression-free survival, overall survival. And now structurally, we can allow for interactions between the genetic variables, between the RNA-seq variables, and in some cases here, the RNA-seq or molecular levels can modulate the clinical phenotypes, and we can look for what's causal for them versus what's reactive. Here's some results from the models themselves, us honing in on specific subnetworks to see what kinds of relationships have emerged. And so one of the strong drivers of response in the system was stem cell transplant, and that's reassuring. So we're learning that relationship directly from the data itself. But But this is a variable and a mechanism that's known to be important for predicting overall survival in multiple myeloma patients. And so some of what we end up learning from these models are known associations as well as new ones, as well as solidifying our knowledge behind what we do know. And so while it's known that stem cell transplant can have an impact on overall survival, we're learning more and more how impactful that can be overall as a first-line treatment for therapy. And so we queried these models to identify other factors that could be driving the endpoint. So we went through and conditioned the model on specific genotypes to specific patients and asked what happens if I upregulate or downregulate all the nodes in the model. And we find then a whole set of targets, potential genes that drive the endpoint. And those classify into different levels. Some of them are known things that have been reported in the literature that now is strongly supported by the models and the data. They also classify into partially known things, what I like to call the known unknowns. So some of these might be targets and mechanisms that have been reported in other cancer types, but we're filtering up as important here. And then some are completely new and novel molecular drivers and targets. Here's an example of another mechanism that was pulled out of the model. It was around the CDK9 pathway and its driver RNA7-SK. So RNA7-SK turned out to be an important driver for first treatment response that lasts at least 1 year before a progressive disease. And while CDK9 and this mechanism was something that was known about, the new association that was pulled up was its link to first treatment response within that 1 year time frame. And some of these insights potentially could help redesign how we look at trials that are looking at that mechanism. Maybe we need to look at shorter duration treatment responses rather than longer ones. There are already clinical trials underway for that target and mechanism, and we're hoping that some of these insights can help drive how those trials are designed or which patients we end up looking in for that. I see that there are some questions that have come up, and I'm going to be done shortly with my presentation, so I will address them in just a second. One of our goals in reverse engineering these models was to create tools that were not just accessible to statisticians that love to query these models in an R or SAS environment, and the models are available to folks in R and SAS, and you can go in and ask what if questions and run your own scripts, your own simulation series, but we wanted them to be accessible to a biologist or a clinician. And so we created a visualization GUI interface that allows someone who doesn't necessarily want a script or know how to go in and interrogate the model. And this is the interface simulation GUI for the multiple myeloma model. So you can go in and a legend can come up and it can show you now all the different variables that are represented in the models, from RNA-seq to clinical variables, various genetic and genomic variables like somatic mutations or copy number variation. You can then hone in on specific parts of this model. So in this case, somebody's querying over JAK1. There are nodes upstream of that, nodes predicted to be downstream of that. All right. And you can pick two things and ask, what happens if I set this node to a specific level? How does that vary or modulate the output? In this case, the user chose these two targets, and then you hit the simulate button. And what the model will simulate is the impact of modulating this node here on that final output node. This is the predicted simulation, and there's a predicted to be an association, causal link between these two based on a simulation. But you can also go back and look at the raw data, so it gives the biologist or clinician an intuitive view as to what is going on in these network models that hopefully they can use and leverage further in their research. Okay. And then a couple more ways that we're using these models. So as I described, we've taken the data and the reverse engineering network models underlying the data, but there's more than one network represented in these models. And the population of models represents two things. The heterogeneity can be due to uncertainty, but sometimes the heterogeneity, especially when you notice certain clusters of set networks emerging, can also be due to the emergence of subgroups and subpopulations within the data set within the study. And so as a follow-on to what we're doing with these multiple myeloma models is we're analyzing them to identify these subgroups as well as potential biomarkers that could classify who are these patients that are more likely to respond to treatments like Revlimid or combinations of Revlimid with IMiD. And then finally, the other use case is really around, once these models are validated in various settings, whether it's retrospective and then eventually prospective, and the final validation stage, as I described with G3, can we then use these insights and these mechanisms to really impact patient care? So as I said, the model itself encompasses those causal mechanisms. You can run simulations and come up with very specific targeted simulations. In the example of the cardiovascular coronary studies, now one can collect new data on these patients, their clinical lab values, imaging, genomics. That can be fed into an integrated clinical IT program, but those new data sets on those new patients can be fed back into the models, and where we can start to use these models to come up with insights that are specific to that patient around their disease progression or what the potentially optimal best treatment is. And leverage some of these findings as well around further targeting around clinical imaging, lab testing, genomics that we may need to add in to get to additional insights. So with that, I think I'm out of my main time for presenting, but do have time for questions, so I'm going to address them now, and I see there's a couple up there already. All right, so the first question, hold on, I think I have to open it up. All right, first question.
Thank you, Dr. Khalil, for that informative presentation. We do have some questions. I'm going to let our audience know how to submit them. If you have a question you'd like to ask Dr. Khalil right now, you can type it into the green Q&A box, push on the green Q&A button in the lower left, put your question in, and hit send. Our first question is, have you applied your analysis to systems that appeared promising enough to launch discovery programs, but ultimately failed clinically? Does your analysis show that the target actually had poor causative power?
Looked really promising that we've then gone in and validated. All right. The next question. Related to my last question, how do you assign a confidence when they're to the primary data used in developing the model? Right. So I assume here, are you talking about the confidence to the actual models themselves or confidence around the data? So confidence around the data itself, we develop error models that can help assign a confidence measure to the data, and that gets propagated all the way through in those Bayesian scores and around the final output. And then the confidence in what we predict is propagated both on the scoring level, and we will try to capture as much of the noise or uncertainty as we can that might, for example, be related to the measurement level, but also propagated to the ensemble where we use the heterogeneity in the ensemble to assess what we're able to predict with certainty. All right, we have another question. Although computational models can be used to predict disease susceptibility and risk, to what extent can in vivo models, especially those that can be rapidly generated using CRISPR-Cas9 technology, accelerate the development and implementation of targeted therapeutics or precision medicine? Right. That's a very good question. So ultimately, in this frame of reverse engineering and then in silico testing out targets, within the framework of the computer model, you have access to every node and every target in the model. And in silico, you can screen many different potential hypotheses, and then the goal is to come up with the mechanisms and the targets that you're most confident in, that you then want to go quickly validate and test. And so I see these technologies around CRISPR or large-scale siRNA screens as further enabling this. Where I would like to see all of this go is where we can set up paradigms of model learning and in silico validations that are very quick and actually quicken the feedback loop between what we're able to predict in silico and what we're able to validate. And in those quick feedback loops, we will generate more data. Every time we validate something, what we learn to be correct or incorrect, or gain confidence in a prediction and less so, and that data can be fed back in. And these models can become more and more accurate and can help us recapitulate more accurately mechanisms underlying our system. So I see these things working in tandem. All right. I did not hear the answer to my first question. Okay. So I will go back to that. All right. So in terms of our systems that appeared promising enough to launch discovery programs, but ultimately failed clinically. So when we've done these analyses, we have found things that retrospectively the model says would not have been a great target, and later or at some point have shown not to work clinically. But we haven't gone as far as applying the targets of this further out in clinical development to see that. But we do see those stories bubble up and pop out of the models. Things that, oh, had you applied this platform and this technology combined with genomics, we could have more quickly either validated something or more quickly failed something. We do see those stories emerge from this. But this is early stages, and we're just finally getting these things launched. And so I anticipate not only will we see more of these mechanisms that either look more promising or less promising that then have either clinical promise or not, but we'll be able to go further in the clinical application of this. Okay.
Thank you, Dr. Khalil. We've had some great questions. That was a great presentation. I want to ask you, since we don't see any more questions in our queue, do you have any final comments for our attendees? Any other information you could pass on?
I urge you to reach out to me directly if you have other questions or would like to have any of this content sent to you. Also, many of the studies described today we have published on, so we can send you those posters and publications as well.
Great. Thank you very much. I want to thank Dr. Khalil for all of her time today presenting this information to us. I also want to thank labroots.com for making this web seminar possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through the end of August 2016. Attendees will receive an email from LabRoots alerting them when this webcast is available for replay, and we invite you to forward that email to your colleagues who may have missed today's live event. Thank you for joining us. We hope to see you again. Goodbye.